In section 17.2, we're going to talk about how populations change or not change based on whether evolution is happening or not. So again, we're looking at microevolution, change that we can actually see in a species over a given amount of time. Remember that we've been talking about the word fitness, that it's the success in passing genes to the next generation. And then we've also talked about adaptation, that makes an organism suited to its environment and increases that individual's ability to pass along its alleles to the next generation. In the last section we talked about single gene traits and polygenic traits. Natural selection can look a little bit different depending on whether it's a single gene trait or a polygenetic trait, multi-gene trait. A mutation in a gene, we're taking a lizard as our example here. A mutation in one gene that determines body color in lizards can affect their lifespan. So we're assuming that the normal color is brown. A random mutation, or a couple different random mutations, can produce red and black forms of that lizard. So let's see how that affects the population. Here's what we see with the different members of the population. We have the initial population here to start out, and you see the frequencies of the different phenotypes. And then we see how that has changed 10 generations in, 20 generations in, 30 generations in. So 10 generations in, still have 80%. All of a sudden, there's no red, and the population of lizards that are black have gone up. 20 generations later, we see the phenotypic frequency has changed. The number of browns are going down, the number of blacks are going up, and then finally, after 30 generations, the frequency of lizards that are black has actually overtaken and gotten larger than the frequency of brown lizards. So what's going on here? A couple different possibilities. For instance, the allele for red coloring might not become common because they're more visible to predators. So if they are able to easily be picked out, they will be selected first by the predators. Their gene will not be passed on to the next generation if they're not given the opportunity to reproduce before they're eaten. What's going on with the black color? Why is that an advantage? We know that black is that color is more easily able to absorb wavelengths of light. So if lizards that are ectothermic, they have to get their heat energy from their surroundings. Higher body temperatures can allow the lizards to move faster, escape predators, and reproduce. So the black coloring might help them maintain homeostasis a little bit better than brown. So we see a shift in that population. Polygenic traits, we have the bell curve back, like we talked about at the end of the last section. The fitness might vary from one end of the curve to the other. So natural selection can affect that bell curve. I'm going to throw you some terms here. Directional selection is the first type of selection. Individuals at one end have higher fitness than individuals at the other, so we see a shift in one direction. We're going to see an example of this in a couple of sections. If larger seeds were available, birds with larger beaks would have an easier time feeding, would be more successful in surviving and passing on genes. So we see a shift to the right. The average beak size increases. The dotted line is where the original beak size was, right here, and we've seen it shift over as generations pass by. So we see a shift in one direction. That's directional selection. Stabilizing selection. If something is kept stable, it's kept the same. So individuals near the center have higher fitness than individuals at either end. Human birth weight is a good example of this. Very small and very large babies are less likely to survive than average-sized individuals. Very small babies, it's difficult for them to survive outside the womb. Very large babies, it's hard for them to get born. So there are complications at either end of the bell curve. The fitness of the smaller or larger babies then is lower than the average-sized individuals. So what you see, again, the dotted line is what it was in the past, more of a spread out situation with birth weight. But over time, you see a shift towards the middle. So that's become more stable. This is stabilizing selection right here. A shift toward the middle. So birth weight has actually gotten less varied over time in humans. Babies with an average mass are more likely to survive. Then we have disruptive selection. If you're disrupting something, you're breaking it apart, you're separating it. So this is when the individuals at the ends of the curve, the ends of the bell curve, have higher fitness than individuals in the middle. So what you end up seeing is the creation of two distinct phenotypes that are separate from one another. It acts against that intermediate, that middle ground. So if you have an area, for instance, where medium-sized seeds are less common, then that would select for the birds with small or large beaks. They would have higher fitness, and the population might split into two groups, one with smaller and one with larger. And that's what you see here. So again, the dotted line is what we started out with. Over time, you see selection for smaller and larger beaks. So these are the three different types of natural selection as it applies to polygenic traits. Genetic drift, just like it sounds, if you think of a boat kind of drifting on the sea or on a lake, it just kind of moves in one direction kind of slowly. Genetic drift occurs in small population. This is by chance. It's a random change in allele frequency. So where do we see genetic drift happening? A couple different situations. We have the bottleneck effect, change in allele frequency following a dramatic reduction in the size of a population. This is usually due to some sort of a natural disaster. So a disaster might kill many individuals in a population, and that would be random. Again, that would be just if it were a natural disaster. This is random. Which individuals, like a tornado or a flood, which individuals are dying off, or in a fire. Sometimes it would be the faster or the heavier or the better swimmers, or in whatever situation would survive. Obviously, it would be a survival of the fittest kind of a situation, but sometimes disasters are unavoidable by even the strongest of individuals. So a disaster can kill the individuals in a population. The surviving population's gene pool might have different gene frequencies from the original. So that's a bottleneck effect. Founder effect, a little bit different. Allele frequencies change as a result of the migration of a small subgroup of a population. That can happen in humans, where you have a group of people that become isolated for whatever reason, whether it's geographically, whether it's based on belief, situating themselves away from other individuals in society. It's a social thing. We have the example on this slide, two groups from a large diverse population of insects. So you have the sample of the original population here on the left, and for whatever reason, you see that the two groups of individuals here have split off from the larger population. So these are called founding populations. Founding population A is made of black and yellow insects, and so you see the descendants being black and yellow. Founding population B, that's split off from the group, is made of only red and yellow insects, and so that group of descendants is only red and yellow. So this is called a founder effect. genetic drift. Little math here. Genetic equilibrium is when there is no evolution. So we're going to talk about the conditions that are required to maintain genetic equilibrium in a population, meaning there's not any change in allele frequency in a population at all. This is called the Hardy-Weinberg principle. And Hardy-Weinberg principle says that 5 conditions are required to maintain genetic equilibrium. Here they are. So the population has to be large. There can be no mutations, which we know is kind of difficult for there not to be any mutations. There must be random mating, so there aren't any selection of mates going on. There isn't any selection based on mating dances, based on shows of displays of prowess, competition between mates. There can't be any movement into or out of the population. You know that is immigration and emigration from your social studies classes. None of that happening. And no natural selection. We're going to go through these one by one. So in order for there not to be, Hardy-Weinberg equilibrium says, in order for there not to be any shift in allele frequencies, and therefore no evolution, there's none of those is going to come into play. So a population is in genetic equilibrium if allele frequencies in the population remain the same. If allele frequencies aren't changing, as I just said, the population will not evolve. So there's no evolution if genetic equilibrium is in play. Very unlikely. So again, Hardy-Weinberg is saying that allele frequencies in a population remain constant, so that equilibrium will happen unless there's one or more of these factors that's causing the frequencies to change. So let's take a look. Large population. A large population size helps maintain genetic equilibrium because you don't have the possibility of genetic drift. There aren't any seals shown. I don't know why that slide said that there were seals there. They're not there. The other one, no mutations. If mutations occur, you have new alleles that would be introduced into the gene pool. And we know, we've talked about mutagens, things that cause mutations. They're everywhere. And mutations just happen on their own. There are copy errors. So in order for Hardy-Weinberg equilibrium to be correct or to be the case about a population, mutations cannot occur. Individuals have to mate at random in order for Hardy-Weinberg equilibrium to occur in a population. We know, though, that there is some sort of selection going on. The example given here, female peacocks choose mates on the basis of physical characteristics. Tail feathers, like I said, displays of prowess, superiority between two males. The female picks the male, like if you're talking about rams. Non-random mating means that alleles for those traits are under selection pressure. Bigger, stronger, faster, like we see here, the different brightly patterned tail feathers, those are being selected for, and so you see a shift in the population. No immigration or emigration. So no movement into or out of the pool, out of the gene pool, because then we're introducing new alleles or removing alleles. And then there's no natural selection. So all genotypes in the population must have equal probabilities of surviving and reproducing. So with our roadkill example, there aren't any animals that are dumber or slower that would be selected against in a population. When we're talking about sexual reproduction and how it relates to allele frequency, it's important to know that meiosis and fertilization don't change the relative frequency of alleles in a population. We talked about that genes are shuffled during sexual reproduction, but it doesn't alter the relative frequencies of the alleles in a population itself, because the shuffling of genes doesn't mean that we're changing the number. They're all there. It's a fixed amount of genes. It's a fixed combination of genes, a fixed set of genes within an individual. They just mix and match and shuffle in different ways in the gametes.